Hello. Thank you for coming. Hope we are ready to go. I'm Seth Crawford. My brother and I co-founded Oregon CBD in 2015. We're Oregon's oldest continually operating hemp farm and the nation's leading supplier of feminized cannabis seed. When I was here last in 2018, I delivered a presentation about the promise and potential of developing non-THC varieties grown as hemp. This work can be seen as an extension of the well-documented breeding program published by Etienne Demeyer at GW Pharmaceuticals, which is itself the culmination of work started at Hortifarm in the Netherlands by Dave Watson and Rob Clark in the 1990s. Our intent was not to just develop clones with novel chemotypes like Hortifarm did. Rather, the goal was to produce field-ready F1 versions that would allow farmers to maximize their yields in a way that brought these cannabinoids into the mainstream. We set out to accomplish that task by applying classical plant breeding strategies to cannabis, isolating individuals and self-pollinating through multiple inbred generations in hopes of attaining higher, higher rates of homozygosity, fixing chemical composition, enabling uniform flowering responses in our hybrids, and attempting to maximize heterosis through careful selection of final parents. We've been engaged in the project for about 8 years now and can report that the first phase is complete. We have CBD, CBDV, CBG, and CBGV parental stock of inbred photoperiod and day neutral lines developed for hybrid F1 seed production. The results of this approach have yielded commercial varieties that set the benchmark for both field productivity and total cannabinoid content in university field trials across the country. Concurrently, they've ushered in a new era where non-psychoactive trim flowers can actually compete on par with their THC counterparts for consumer attention. Farmers have received over 40 different awards with our varieties around the U.S., including a really cool landmark win at the Cultivation Classic in 2020 by Horn Creek Hemp, taking the blind tasting flagship category of most credible cultivar with our CBD-dominant sour space candy. This beat out a field dominated by THC specimens because of its consistently relaxing and positive effect. We're a small R&D operation with an oversized influence on the hemp market, in large part due to our attention to detail in the breeding process and our desire to collaborate with other scientists to better our collective understanding of this incredible plant. We've built out facilities that include over 275,000 square feet of next-generation indoor greenhouse space, 600 isolation tents for controlled crosses, a tissue culture and ploidy manipulation lab, whole genome sequencing and molecular lab capabilities, and full chemical analysis at our little 49-acre farm in Independence, Oregon. We've basically been lucky enough to build a professional life that the 20-year-old version of ourselves couldn't have dreamed up, and we're tremendously thankful to have been born in the right place at the right time. Who knew that a couple kids raised in an electricity-less log cabin in the woods of Southern Oregon would get such an amazing opportunity? So big thanks to Mom and Dad. I'd also like to thank the entire CannMed event team for hosting what is, to me, the most outstanding cannabis conference in the country, and thank all of you for taking the time to listen today. Many of the best and brightest minds in the cannabis space are here, and we're all very lucky to get firsthand accounts of the work being accomplished right now. I'd also like to thank everybody on our whole team at Oregon CBD, past, present, and future, for their contributions to the work that's going to be discussed today, and say thanks to all of our collaborators over the past few years. There's too many to mention right now, but they're going to be acknowledged throughout this presentation. So the title of today's talk is phrased as a question: 2022, the golden year of cannabis? Full disclosure, it had been 2020, the golden year of cannabis, and then a couple years of epidemiological history got in our way. I want to address the question at the outset so everybody's able to concentrate on the rest of the talk. Yes, this is the golden year. Last year was too, and the year before that, and next year probably be as well. It can be true regardless of your worldview, whether the glass is truly half full or totally empty. As long as we can grow for another season and build on what we collectively learned last year, it's always going to be the golden year. All dialectical meanderings aside, we do stand at an absolutely sublime moment in time for cannabis research, in no small part due to the rise of hemp in the U.S. The general framework for today includes a brief overview of that program, a more in-depth dive into the use of ploidy manipulation as a breeding tool, with triploids as the primary example. And finally, we'll finish up with some thoughts about the next phases double entendre there on purpose, genomics and plants that will push our industry forward. There's going to be ample time at the end for both public questions, and we do have a break conveniently scheduled afterwards, so if it doesn't get addressed in the public comment section, please come find me at the end. I'm happy to answer anything. The U.S. Hemp Timeline. So industrial hemp was federally legalized in the U.S. via the 2014 Farm Bill, passed in December of that year, with a maximum allowable THC limit of 0.3% or less. With the late passage, many states took a number of months to adopt conforming policies and start issuing licenses to growers, making 2016 the first production year in any meaningful sense. A strange patchwork of state-enforced rules were the name of the game from then until this year, which is the first to be conducted under final USDA rules. These gray market area years led to variable sampling methodologies, compliance test timing, and reporting requirements across the country, with significant confusion amongst regulators and farmers alike, occasionally leading to whole crop destruction when plants tested over the THC threshold. As has been established by many, this THC limit is demonstrably arbitrary and not grounded in science, but that's what we have to work with until Congress updates the rules. The biggest watershed moment on this timeline occurs at the end of 2018 with passage of the most recent Farm Bill, which did away with the research pilot aspect of the original law and placed fully legalized hemp production in the U.S. under the jurisdiction of the USDA moving forward. USDA issued an interim final rule for producers on Halloween of 2019. It was definitely a scare. Proposed that sampling occur within 6 days of harvest and be taken from the top 2 inches of terminal flowers. So basically, no economically valuable CBD varieties that were being grown in the US at that time would pass testing. The public comment period was intense. Farmers were pissed. And luckily, this time around, we had some help from universities studying the crop for the first time. After reviewing public feedback and coordinating with other affected federal agencies, the USDA concretized the final rules on January 19th, 2021. These require that all states with hemp programs submit a control plan for USDA approval, which most have completed at this point. Would-be producers in states without formal programs are able to apply for a license directly from the USDA. The final rules made compliance sampling much more uniform nationally, with a 28-day pre-harvest window instituted, and a sampling protocol that calls for the top 5 to 8 inches of the main female cola. While some variability still exists from state to state and from sampler to sampler, it's been tamped down substantially through this harmonization process. In a parallel effort, the National Institutes for Standards and Testing started a voluntary program for cannabis testing labs in May of 2019 called the CANA-QAP, or Cannabis Quality Assurance Program. The goal of this program is to establish uniformity in analytical measurement through distributed testing of material with known cannabinoid profiles and publicly releasing the results. Taken together, it's clear to see that federal agencies are preparing for national legalization, and hemp is serving as the test case. From 2020 on, a number of previously unavailable cannabinoids entered the marketplace. So the Delta-8 THC, Delta-10 THC, THCO, hydrogenated cannabinoids. If you can name it, enterprising chemists with varying levels of skill and equipment were making it out of hemp-derived precursors. While there's been significant consternation about the safety of these products, largely due to residual contaminants and unknown compounds being generated in the conversion process, movements by individual states to rein in potentially intoxicating hemp derivatives have really been met by a lot of permissiveness by the with the federal response. The last note on this timeline directly addresses these new compounds and essentially any other product made from cannabis grown as hemp. On January 6th of this year, the DEA sent a policy determination letter to a hemp product manufacturer that clearly stated the agency does not have enforcement authority over hemp in any way. Delta-8 THC including Delta-8 THC derived from the isomerization of CBD. So this obviously has significant implications. Production of hemp over this same period of time underwent an absolutely meteoric ascent, peaking in the summer of 2019 following passage of the new Farm Bill. Overproduction ensued, prices plummeted, and inaction by the FDA created a cacophony of doubt amongst established product manufacturers over the future of hemp-derived cannabinoids. The initial rapid expansion in production was met with an equally precipitous decline in total acres registered and farm gate prices, to the point where it's not economical to grow for most farmers. Biomass prices, so this is the floral material destined for extraction, bottomed out around 50 cents per pound earlier this year and are slowly creeping up as the tremendous glut of material finally starts to dissipate. The trimmed hemp flower market is still holding on, but is a very small proportion of the total acres planted each year. Registered acres, as you can see, in 2022 are currently estimated to be slightly more than 2017, but still quite a bit less than 2018. The few farmers who have held on over the last 3 years are desperately hoping for a solid price rebound, but at this point, who really knows what's going to transpire? If anything, the tendency of boom and bust cycles with recently opened cannabis markets is a stark reminder that efficiency, marketability, and novelty are all mandatory for growers to simply stay in business. The silver lining here, despite the market collapse, is that hemp, viewed as a vehicle for research, has absolutely exploded during this production collapse as universities joined industry in their ability to handle the plant in a relatively uninhibited way. A key regulatory, scientific, and practical farming question has been, where does residual THC come from in type 3 and type 4 plants? The question was of critical importance while the USDA was proposing rules for the hemp industry, as their initial versions would have essentially ended CBD production. And we believe in this case that some good science actually affected national policy for once. Onofre and co-authors gave us some tantalizing clues in their 2015 survey of GW Pharmaceuticals' diverse germplasm library, demonstrating a relationship between cannabinoid synthase variation and quantitative chemotype. In essence, certain single-letter nucleotide changes in the genes responsible for CBDA and THCA production lead to different cannabinoid purities, ranging from 66 to 90% in the case of CBDA in type 3 plants, leaving up to 10% of the total cannabinoid fraction as other compounds, even in the purest CBD producers. Zerpel's 2018 paper demonstrates that CBDAS, the gene responsible for CBD production, in vitro produces CBDA, THCA, CBCA, and 5 other unknown compounds in ratios affected by substrate pH, but reportedly normalized to 20:1 for the known products. Larry Smart's lab at Cornell has done some great work over the past 3 years cataloging and characterizing commercially available hemp varieties and getting great graduate student publications in the process. Their work on time series cannabinoid accumulation in field trials shows that most type 3 germplasm grown in the US tested about 25:1 CBD to THC throughout its flowering period. Taken together, we now know that most THC produced in these high CBD varieties is coming from the gene responsible for CBDA production. You can't have one without the other, but their co-presence is predictable. During this time, we spent a lot of energy in these intervening years developing varieties that would meet a stringent 0.3% day of harvest limit, since that was the initial direction proposed by the USDA. We were successful in doing so with CBG-dominant varieties as well as CBDV-rich varieties. Our CBG varieties are made possible through a synthase variant we found in one of our inbred populations. It's a full-length THCAS with a single SNP at 1,064 base pairs, responsible for significant loss of function, with final flowers averaging 100:1 CBGA to THCA ratios, and always compliant when grown as hemp. Our work with high varin content varieties has shown that, unlike their pencil counterparts, the cannabinoid fraction undergoes dramatic changes over the course of flowering. The varin proportion of total cannabinoids decreases as flowers mature. From a compliance perspective, this is a really beneficial outcome, since THCVA is not included in the 0.3 total THC harvest limit. In this example, THCVA is present at levels 3 times higher than THCA at that critical 28-day pre-harvest window, but the two drop to an equal 1:1 ratio by the end of flowering. The shift in federal policy to a 28-day pre-harvest test allows nearly all type 3 plants to test below the legal limit if varieties are fully mature in 8 weeks, even with high cannabinoid content hemp. And we believe that this is a good compromise until Congress can be convinced to implement a 1% total THC rule across the board. Other recent research has significantly contributed to our understanding of specific traits and relative genomic location. The Grasso-Weiblin team's excellent publication accompanying the CS10 reference genome release found total cannabinoid potency markers present on chromosomes 3, 4, 6, 9, and the X chromosome. There have also been some really great papers on terpene synthase pathways recently with the Allen, Booth, and Zager research, 3 independent papers, really standing out as the most useful. These help us place many of the mono and sesquiterpene TPS variants on chromosomes 1, 5, and 6, with some diterpenes on chromosomes 4 and 7. The Van Velzen paper on cannabinoid oxydocyclase gene families places the CBG-AS synthase on the X chromosome as well. Our team used Lighthouse Genomics SNP chip data from a segregating F2 population to co-publish with Dewey Scientific and identify PM1, the first powdery mildew resistance gene in cannabis, and show that its location is on chromosome 2. Our Eurofin SNP chip validation run on a different F2 population containing day-neutral individuals placed the mechanisms controlling that particular flowering response on chromosome 1 as well. Subsequent Illumina shotgun sequencing of the same population by INR gene and data analysis with our collaborators at OSU and the Vining Lab yielded markers associated with elevated total propyl cannabinoid fractions, which we know from other publications to be a polygenic trait on chromosomes 3, 4, 5, 7, 8, and 9, with a single marker on chromosome 3 having the greatest impact. Between the shifting policy landscape and research developments, cannabis essential oil breeding programs are quickly moving from an opaque guessing game requiring multiple methods of triangulation to one where many traits can be determined at the seedling stage with targeted PCR tests, and particular crosses can be designed in a way that maximizes successful outcomes while not getting mired down in impossible dreams. This becomes even more important as we consider that the frontier of production of all cannabinoid plant types will most likely be polyploid and much more genetically complex than the diploids we all know and love. So why triploids? Let's turn to triploids. Triploids are cannabis plants that are basically incredibly resistant to pollination, as well as have exciting attributes brought on by extra genome copies. Our development of triploids was born out of the trauma of losing several crops to cross-pollination events in 2016 and 2018, and hearing horror stories from growers year after year. If you've grown cannabis, you likely understand how devastating pollen drift can be on crops, and that it can travel hundreds of miles in the right conditions. Fully pollinated plants produce 50% less flower weight and 30% less cannabinoids. A seeded crop is essentially worthless if you're growing for trim flowers, and still devastating if your end goal is biomass and extraction. As we've also seen in the first 7 years of production in Oregon, feral populations of cannabis are regularly establishing themselves in fallow fields and have become a big enough concern that our Department of Agriculture is now working on rules and penalties for landowners who fail to control rogue plants. In newly opened Midwestern production areas, previous research has shown that over a third of all airborne pollen in the summertime in the month of August is derived from feral hemp. Having a plant that's relatively incapable of being pollinated could fix a lot of those issues and threats and let farmers put their energy into growing great crops instead of worrying. So to understand triploids, we need to establish some basic terminology, and I apologize to anyone who this is easy. For me, it is not. It's been a long trip. Chromosomes are packed together collections of DNA. So DNA is the underlying genetic instructions for life. It's helpful to think of chromosomes as books of code, which make up the library that is an organism. Every species has a unique number of books in its library. Humans have 46, naked mole rats have 60, and cannabis has 20 in the diploid state. This library size is referred to as the chromosome set number. Ploidy refers to the number of homologous sets of chromosomes in an organism. Organisms with a single set are referred to as haploid. 2 is diploid, 3 sets triploid, 4 sets tetraploid, so on and so forth. Any organism with a ploidy higher than 2 is referred to as a polyploid. In sexually reproducing species, 1/2 of the chromosome sets are inherited from each respective parent. For humans, this means our individual genome is actually a fusion of two different genomes cut in half during reproduction. We're truly one half of both parents. The same is true for diploid plants. While most vertebrates are diploid, many plants are polyploid. These include major agricultural crops of critical importance to humans, such as coffee, bananas, strawberries, bread wheat, oats, citrus, potatoes, and tobacco, to just name a few. The main point to understand is that polyploidization is a natural phenomenon that has occurred many times in nature and has led to the creation of the ancestors of many modern crops. Polyploid induction in nature tends to correspond with extreme climate events or natural disasters and lead to increased diversity within a species. The only documented naturally occurring tetraploid cannabis population was found at 14,000 feet in elevation in the Himalayas by an Indian research effort about 10 years ago. The extreme climate and altitude may have been the causal agent responsible for that particular event. We can think of this as nature's way of creating options for diverse responses under changing circumstances. It's also important to note that these events are not changing the underlying DNA in any way or introducing new genes. They're simply whole genome duplications of what was already present. In the case of polyploids, more gene copies is believed to confer better adaptability and resilience. Plant breeders became aware of the remarkable superiority of polyploids in the mid-20th century and have induced polyploidy and or used natural polyploids in many ways to obtain increasingly improved plant cultivars. Some of the most important consequences of polyploidy for plant breeding are the increased size of plant organs, so bigger flowers, bigger leaves, bigger stems, the buffering of deleterious mutations, increased heterozygosity, and superior heterosis or hybrid vigor. Cultivars have been generated with higher yields, improved product quality, and increased tolerance to both biotic and abiotic stresses. Polyploidy is a tried and true method of improvement in many crops. An important bonus to all this is that plants with odd chromosome set numbers, so triploids, pentaploids, and so on, often prove to be infertile. This is what makes seedless watermelons, bananas, hops, and now cannabis a reality. The only thing you need to know about this is that the haploid cannabis genome is 800 megabases in size. We'll save some time. Our polyploid development program was designed and led by Dr. Shen Chen, OSU grad who's now an assistant professor at NC State. Our goal was to convert our diploid breeding stock. And these take 3 to 4 years to develop on their own. But our goal was to convert these into tetraploids and beyond, test the fertility of various ploidies in controlled environments, and to test the agronomic traits of various ploidies via controlled crosses and initial field trials. This work began in early 2019 and was occurring simultaneously to the efforts at Canopy Growth Corporation and Jessica Lebel-Brand's lab at the University of Connecticut, both of whom have recently published papers describing their processes, and we did as well. I say this as a way of not so subtly conveying some disapproval of the most recent claim to emerge from a large purveyor of clones here in California, who, if you were at the Emerald Cup last December, you probably know what I'm talking about, just launched what they purport to be the world's first triploid cannabis. In a day and age so rife with misinformation, history will probably not care one way or the other, but for some of us, truth is more important than marketing efforts, and I for one, believe it's important to acknowledge the labor of others that led to increased knowledge that benefits us all. On that note, while our company was actually the first to offer triploid cannabis commercially in 2021, the Hortifarm group that I had previously mentioned, led by Dave Watson, were likely the first to test triploids back in the 1990s, and I'd like to publicly acknowledge that effort and his encouragement of our program as we went through the R&D phase. So that aside, let's get into the details and our findings from the past 3 years on cannabis ploidy research. Controlled genome doubling events require initial plants, a cell division inhibitor, steady hands, patience, and a flow cytometer, which is a really cool piece of equipment capable of measuring an organism's cell size. Hundreds of asexually propagated clones of a target plant genotype are pruned back until a single growth point remains, and then colchicine is applied to that growth point. Most plants die within a month. The few survivors are screened by flow cytometry, and any confirmed tetraploids move on to the stabilization process. At each step, plants are rescreened repeatedly, as they have a tendency to revert to lower or mixed ploidies until fixed. A term I use lightly, as diploid reversion is always a possibility under stressful conditions. If a plant has 10 shoots, all 10 shoots are tested. This continues until that relative stability is attained, and then the plants are ready for seed production in our case, but not before a final flow testing. To date, we've now successfully created tetraploid versions of all of our classic CBD, CBG, CBDV, and CBGV production mothers, as well as a long list of type 1 plants in our germplasm library, and have incorporated the streamlined conversion process into our breeding program. Start to finish, this process takes about 8 months. It's not the breeding that leads up to it, but just the conversion process is about 8 months. If you guys have time, everybody have time? We've got about a 3-minute video clip. Okay, cool. Everybody's got time. This is Brendan Rojas. He is our Ploidy lab manager, and he's going to walk you through a very quick description of what it looks like in the greenhouse and the lab to actually convert and test these plants.
I'm going to go through the process for converting plants. The first step is, after we've gotten the plants, is we'll trim it down to just a single meristem. For this plant, you can see it's right at the top, just right here, this one single meristem. And after that, what we'll do is we'll take a piece of cotton and we'll just wrap it right around that one single meristem, and then come in with colchicine and add just a little bit of colchicine to fully hydrate that cotton swab. So after everything's been treated, we need to keep the plants moist and warm so that they have a good environment to grow in. To do that, we use a second one of these black trays, and we prop it up with cinder blocks, and then we use this plastic to wrap all the way around to ensure that it holds that humidity inside. Once everything's been treated, we do the treatments for 4 days in a row. At the end of the 4th day, we pull the cotton off and then rinse the meristems with water so that we rinse off any of that excess colchicine. And then after they've been treated, in about 2 weeks, we'll start to see growth from those points. And with that growth, we'll take them up to the tissue culture building to start running flow on them and to check our results. So at this point, I've got a plant that's been treated with colchicine. Just one single meristem was treated, and then what we have in front of us is a plant that's grown up from that single meristem. And what I'll do is I'll come in with a pair of scissors, and I'll take just one leaf sample. The samples can range in size. We don't need a whole leaf. I can use as little as just a leaf tip, very small. We have 2 buffers that the sample goes into. The first one's an extraction buffer. That just helps make sure that when you're chopping the plant material, the nuclei in the plant material don't break apart. And the second one is a fluorescent stain buffer, which dyes the nuclei of the cells within the plant material, which makes it possible for our flow cytometer to actually read the genetic material in the nuclei. So this is the extraction buffer. It goes into a petri dish. It gets chopped. The nuclei get stained. We then run the plant material through a filter so we don't clog up our machine. It goes into the sample cell, and then it should start running. So after the sample's been prepared and we put it into the flow machine, what the flow cytometer actually does is it takes the liquid that we just prepared and it runs it through a very small cuvette in a single file line where it lines up all the nuclei in just one row, and then it shoots a UV light through the nuclei as it goes through this one cuvette. So based off of how the UV light reflects off of the nuclei that we just prepared, that's how it gets the readout, and we get the results on a graph. So it's kind of like a relative peak of the genetic material within the nuclei.
Awesome. Thank you, Brendan. It's Oregon cannabis breeding, duct tape, zip ties, and now with laser beams. Our lab's been operating with the 2 Quantum P flow cytometer units for the past couple years. We just recently added a high throughput Cytoflex V2 with 2 lasers, which I'm told is great. It's higher throughput, allows us to use standard 96 well plates to be able to have a much larger number of plants get screened at any given time. You can see in these images the resulting peaks generated by that flow cytometer when testing plant material of various ploidies. So flow cytometer results on the far left, and then some chromosome squash pictures from microscope in the middle there. Using multiple methods to validate ploidy levels is especially necessary when creating baselines for further analysis, but the chromosome pictures here help to convey the cellular changes present in these polyploids. There's really a lot more genetic information in these plants, and you can see it when you get close enough. Diploid cannabis plants contain about 60,000 genes and have a genome size of 1.6 gigabases. Triploids contain about 90,000 genes and have a genome size of 2.4 gigabases. Tetraploids contain 120,000 genes and have a genome size of 3.2 gigabases, or roughly half the size of humans. These extra gene copies are what is believed to drive resilience and adaptability in polyploids. So seedlessness in triploid cannabis, that'd be an awesome thing, right? But we had to design some experiments that would adequately test that claim. To do so, we created 6 crossing combinations to assess seed production and viability. Diploid, triploid, and tetraploid versions of the same F1 hybrid were pollinated by diploid and tetraploid pollen donors. These trials were conducted in sealed greenhouses, and the target plants were subjected to massive amounts of pollen. This is intended to simulate a flower crop grown side by side with a large grain farm or dense feral populations. The results show that diploids pollinated by diploids produce about 2 times as many seeds as tetraploids pollinated by tetraploids, suggesting that ploidy increases result in lower fertility in cannabis. Tetraploids are not viable as a pollen donor for diploids, but are able to induce many problematically empty seeds. The key finding is that seed production was essentially non-existent in triploids when pollinated by either a diploid or a tetraploid in this experiment. Even under substantial duress, they remain nearly seed-free, with a maximum average of 6 seeds per triploid plant when pollinated by diploids in the controlled pollen inundation zone. In addition to significantly impeded fertility, triploids also demonstrated superiority in many key agronomic traits relative to their diploid counterparts. We conducted outdoor field trials at our campus in 2020 with several different diploid, triploid, and tetraploid crossing combinations. Flower production increased between 30 to 100%, although that was variety dependent, with the largest leap seen in day-neutral varieties. The yield differences are statistically significant, but the visual differences in plant size, larger leaves, larger flowers, thicker stems, denser trichome coverage, tells you all you need to know about triploid vigor. There's a slight increase in total cannabinoid content in flowers and a marked increase in, in trichome density on sugar leaves near flowers. Additionally, olfactory compounds were noticeably more abundant in triploids and tetraploids compared to diploids, but we did not test for total terpene content in this study because terpenes tell us less than half the story, and most of the major aroma components of our varieties are not terpenes at all. There's more about that to come. We don't have enough time for the entire story today, but we'll get into it with a little bit more detail later. These early results suggested that triploids are much more resilient to any challenges they face relative to diploid counterparts. In 2021, we followed up with extended field trials at North Carolina State University, Cornell, and the University of Kentucky as part of our founding membership of the FAR-sponsored Hemp Research Consortium. The study design at all 3 institutions were very similar, with diploid and triploid versions of our feminized varieties planted on 4-foot centers at 2 separate outdoor field sites. In each study, one field was designated as a negative pollen control, and the other was purposefully planted at a high density with male-female diploid grain varieties immediately adjacent to the trial locations. Final research reports from all 3 institutions will be available on their extension website soon, but they were kind enough to grant us some of the preliminary data for today. In North Carolina, the Goldsboro location served as the positive pollen environment and Salisbury the negative control. White CBG, Suver Haze, and Lifter varieties were trialed in both diploid and triploid form. Thanks to David Suchoff and his team for their work on this trial and the subsequent data. At the negative pollen control Salisbury site, differences in biomass, defined as non-stem, flower, foliage, and any seed produced, Differences in biomass between the two ploidies at harvest was not statistically significant, but it is worth noting that the amount of variation was greater in the triploids. At the positive pollen Goldsboro location, biomass production was greater in triploids by about a quarter pound per plant, and the difference was statistically significant. Differences in seeding were all statistically significant. Pollinated diploids produced about 2,500 seeds per plant, while triploids were around 50, or about 2% relative to their diploid counterparts. Seed weight per plant followed a genotype-specific pattern that at this point generates more questions than answers. Relative seed size between the genotypes varies, with lifter seed much smaller than white CBG in general, which may account for some of the variation seen here, but there also seems to be an unexplainable element at play as well. Note the single outlier with much higher seed weight present in the triploid Suver Hayes variety. It's just a little line up there on the triploid section on the far right. Trials at Cornell included lifter, Suver Hayes, and white CBG in both diploid and triploid forms, as well as sour lifter, sour Suver Hayes, and sour RNA as triploids only. Thanks to Larry Smart and his lab for their work on this project. This particular trial is a great example of the phrase, farming happens. Strip biomass yields highlight this, as triploids did not fare as well compared to their diploid counterparts, with Suver Hayes being the lone exception. The problem here was that large portions of the negative pollen trial site were subjected to catastrophic flooding multiple times this past summer, and we're eager to follow up next year to get improved data. That said, it's definitely an example of a time where negative environmental events do seem to impact triploids more than diploids. In terms of seed counts and weight, triploids again demonstrated their remarkable infertility relative to diploids. At the pollen positive site, triploids contain an average of 11.3% as many seeds as diploids and produce 10.5% as much seed weight as diploids. While a good result for triploids, the seed amounts are significantly higher than those reported at NC State. In Kentucky, the North Farm location served as the positive pollen environment and the South Farm the negative control. In this study, lifter and white CBG were trialed as diploids and triploids, while our new Forbidden V and Pine Walker CBD V dominant triploid varieties were also included. Thanks to Bob Pierce and his team for their work on this trial and data collection. Biomass and seed yields were reported as pounds per acre in this study. With the exception of diploid lifter, overall biomass yields were higher at the pollinated site relative to the negative pollen control site, which, when taking the underlying seed weights into account, suggests that environmental conditions may have contributed to this particular outcome as well. That said, triploids consistently produced more biomass than their diploid counterparts in both locations, and again produced very few seeds when subjected to pollen challenge. One of the interesting, unanticipated pieces of information that we received from the Kentucky trial is some observations on relative disease resistance between diploid and triploid cultivars. During the 2021 growing season, University of Kentucky observed that triploid versions of lifter appeared to be significantly more resistant to leaf spot diseases than their diploid counterparts, as measured by the percentage of the leaf affected by the disease. This pattern of resistance was true for 3 of the most important foliar fungal pathogens observed in the southern region of the United States: Cercospora, Bipolaris, and Septoria. These 3 pathogens, each of which cause leaf spots where spores land on the leaf surface and colonize the surrounding tissue, are some of the most economically important pathogens of the southern part of the country. Their relative importance to each other is still being investigated, but as you can see in the photograph, they have the ability to cause major reductions in economic yield and likely exist in disease complexes whereby several of the pathogens are present at the same time causing disease. The same trend was not seen for white CBG. The diploid and triploid counterparts were approximately equal in terms of their susceptibility to these particular pathogens. Furthermore, while we don't have diploid comparisons for our CBD-V cultivars, those are all triploids, they appear to be extremely resistant to all 3 fungal pathogens. Increases in ploidy have been implicated in increases in disease resistance in other cropping systems, for example, in hops, the most closely related plant of economic importance to cannabis. Research has shown that the effect of ploidy on disease resistance can be highly dependent, and it's dependent not only on the crop species, but can vary even amongst varieties within the same species. In this way, it's not surprising to see this impact of Ploidy on lifter and not white CBG. Kevin McKiernan's team, in their preprint a couple years ago, noted an interesting correlation between CBCA-S cassettes on chromosome 7 and disease resistance in cannabis plants. And there's still a lot of inquiry into that line of reasoning. But what I wanted to mention is fairly interesting and not entirely proven at this point, but our CBDV dominant varieties have basically 4 copies of that CBCA-S cassette over and above what a normal type 3 plant would have. So it's a fairly interesting correlation. These initial data from Kentucky help give us important insight into disease resistance in cannabis and how we may start to leverage this information for future breeding goals to make more robust varieties for farmers. The farmer experience in 2021. The quote at the very top sort of says it all. Everything else we grew last year got seeded. It was kind of the experience with folks that were growing triploids and diploids side by side that underwent pollen challenges, with a few exceptions. Indoor farmers relayed nearly universal acclaim on performance, especially when subjected to unplanned pollen challenges. They report qualitative improvement in overall aroma character and intensity, faster maturation of flowers, larger yields, and probably most importantly, dense top-grade flowers even on lower branches. The main drawbacks reported are that triploids tend to perform worse than diploids in low light conditions. So again, another situation where the environment ends up impacting plants that have higher ploidies in a little bit more profound way. There were also a small number of isolated, cosmetically undesirable hermaphroditic flower sports on these triploids compared to the diploid counterparts. Farmers' outdoor experiences were very positive, and relative triploid infertility seemed to prevent seed formation for most, including several in Colorado with intensive feral hemp populations that have been decimated the last couple years. Outdoor flowering response was slower to trigger by about 2 weeks, largely driven by the underlying genome imbalance of our hybrids. So 2 genomes are derived from the photoperiod parent and 1 from the day-neutral parent. The opposite is true when the pairing is reversed. So 2 day-neutral genome copies and 1 photoperiod genome copy leads to earlier flower formation, with a late August, early September harvest time at 45 degrees north in the experimental varieties we trialed last summer. In general, farmers loved triploids, and many swear to never grow diploids again, and honestly, myself included in production environments. There were several outliers worth noting, because as awesome as these higher ploidy plants are, nothing is a cure-all. We had reports from about 12 farmers out of hundreds last year who were seeded at levels that were higher than sporadic. Not as bad as diploids under full pollination, but more effective than we had previously seen, including at the university field trials. So Imre Garcia, our client support expert, and Andrea Garfinkel, the director of R&D for us, visited most of these farms in person, collected samples, and got as much detail as possible from the farmers. Leaf samples from the affected plants were confirmed as triploids by flow cytometry. Some seeds were capable of germination, this is pretty interesting, and tested between diploid and triploid, making them alloploids with more than 2 sets of homologous chromosomes, but less than 3. The pollen pressure impacting the farms were equally mixed between very large feral populations in the Midwest and the surrounding areas, intense localized pressure from neighboring farms, and what we termed paranormal triploid self-pollination, where no other discernible explanation could be identified. Continued field trials and data collection are necessary to better understand these anomalies, but I think it's safe to say that they're definitely outliers from the overwhelmingly positive results most farmers experienced. But definitely not the only anomalies we've seen. This plant just never ceases to amaze. Small case in point. In the summer of 2020, my brother Eric picked out 4 absolutely outstanding plants in a highly inbred diploid population of day-neutral type 4 plants grown from seed in our breeding program during a seed multiplication run. This is a parallel breeding line to the pollen donor that we use to create white CBG and stem cell CBG. This is going to be the final selection out of about 3,000 plants. These were literally the best. And you know what's going to happen here, right? The plants had larger leaves and flowers, more resin content, and far stronger aroma than any of the others. Ploidy screening revealed that all 4 were tetraploid, while the remaining population were diploid, as expected. Seeds produced on these tetraploids were screened, and they came back as triploid. Why were there tetraploids in this population of diploids? Fast forward to last spring. We submitted our first commercial triploid seed for germination, purity, and ploidy screening to the Oregon State University Seed Lab and received these results. Relative rates of triploidy in seed produced from tetraploid seed carriers and diploid pollen parents appears linked to the pollen donor lineage. In this example, Suver, Hayes, and Lifter, both of which share the same ERB day-neutral pollen donor, tested at 100% triploid. The sour versions of both, which utilizes the same tetraploid production mothers, but switches pollen donors to a unique terpinolene-dominant day-neutral variety, resulted in 97 and 93% triploidy rates, respectively, while the remaining 3 to 7% tested as tetraploids. So I guess we were also the first to release tetraploids. Didn't really mean to do that, though. The same is true in stem cell CBG and white CBG varieties, where 1 to 5% respectively tested as tetraploids. So armed with this confusing new knowledge, we implemented a new step in our commercial seed production program this year, where every pollen donor was ploidy screened to confirm diploid status. While no tetraploids were found in any populations, triploids were present in both donor lines at rates less than 0.5%. Even with the offenders removed from our process, the resulting seed continues to throw low rates of tetraploids in certain genotype combinations. It's just fascinating to me. This is made even more confusing when the breeding history of the pollen donor is interrogated. These are lines produced via multiple generations of self-pollination of single plants in isolation tents with HEPA filters that only I have access to. Like, I made all these seeds. I know with 100% certainty, and we confirmed it via flow cytometry, that the parent was a diploid. We've done whole genome sequencing on both the parent and resulting offspring. We know that these plants are from the same source. There's no good explanation. What we've received from Oregon State University is we've been told that it's, I consider it a speculative alternative, related to a non-disjunction during meiosis. Like, okay, I know that has something to do with pollen and cell division. There's got to be a fascinating dissertation for someone in there to figure out what's really going on. But what I'm absolutely awestruck by is that nature does find a way. In this case, the first triploid cannabis was actually released by cannabis itself, probably millions of times over, and long before humans existed in modern form. So it's a good one for cannabis. In summary on triploids, we're seeing dramatically reduced fertility, although not 100% seedless. It's just, that's the reality. It's very close, but not quite. Relatively unchanged cannabinoid content between diploids and triploids and even tetraploids, although there is some evidence that suggests that there's a slight increase, and I think we need to do more replications of that to get to the bottom of it. In our experience, yields are higher, but come with that slightly delayed flowering response. The most dramatic example of yield can be found in our day-neutral varieties. Our best 2018 day-neutral diploid yielded about 250 grams of flower, which is really good for a day-neutral. This is an auto-flowering variety. Very good. But the improved triploid version this past summer produced over 658 grams of usable flower. And my brother did not change sizes between those two pictures. Lost a little bit of hair with two newborns, but same size guy for scale. Olfactory compounds seem to be qualitatively heightened. And I say qualitatively, this is a sticky issue, likely due to combinatorial effects of unique aroma pathways being multiplied. We don't have enough time today to get into the details, and it's not my story to fully tell, but if you want breadcrumbs, do search for unfunded USDA grants on hemp aroma compounds. But the general point is that terpenes contribute about 50% of the total aroma in cannabis. Most analytical reports on them are not very helpful because there's only 8 to 10 terpenes, depending on the variety of cannabis, but about 8 to 10 terpenes that are even above the detectable odor threshold. So when you're looking at a terpene test result that has, 0.05%, depending on the compound, you may not be smelling that. And so a lot of the analytical data that we see commercially available for folks isn't actually capturing what's responsible for those underlying aromas. The rest of the aroma is comprised of things like esters, sulfur-containing compounds that were described earlier today, aldehydes, and others that can dramatically impact the sensory experience even when present in trace amounts. We're talking like parts per billion. The hypothesis here is because unique precursor pathways are responsible for many of these compounds, their presence and diversity could be driving the overall loudness of aroma, which would then be magnified by increased copy numbers in polyploids. There's obviously much more work to be done in this area, but the early findings lend support to this view. There's obviously some big mysteries to be figured out regarding pollen ploidy and identification of cannabis's most efficient ploidy level for various production purposes, but our work to date strongly suggests that lower ploidies, so haploids and diploids, are best for breeding work, while higher ploidies, triploids and tetraploids so far, with pentaploids and octoploids to be tested next, seem to be better suited for commercial production. I'd like to wind down today's talk with some yet to be completed research that's near and dear to my heart and what I personally believe will be one of the lasting legacies we can leave to future generations of cannabis breeders. New advances in sequencing technology and bioinformatics pipelines are allowing for the deepest, most accurate exploration of organisms that we've ever seen, to the point where the data is actually pretty useful and reasonably priced relative to the return on your investment. In our lab, these advancements allowed us to shrink our computing needs from a 384-core computing cluster the size of a negative 80C freezer, pictured here, down to a well-equipped laptop. The wall time for assembly has gone from over a week to just under 5 hours. The quality of the assembled genome has improved from synthetic haploids to partially phased diploids using a PacBio SeqWell2 and the freely available Hi-Fi ASM assembler. The order of magnitude increase in efficiency and highly accurate reads are moving us from an era of a single reference genome serving as a representative of an entire species to one where the full diversity of a species can be represented through hundreds or even thousands of reference quality assemblies. This is the new era of pangenomes. Important agricultural crops like corn, soy, cotton, and apples are some of the first plants to undergo this process. Fortuitous historical circumstances have made it possible for cannabis to be brought into the club as well. Greg Concepcion of PacBio recorded a talk after the cancellation of PAG this year, where he outlines the basic steps of pangenome construction and Pangenome tool application in plants, using cannabis as the example species, and our genome collection as the initial library. If you haven't seen it yet, please check it out. Greg's been contributing to the cannabis genome research efforts for years now, and we're all really lucky to have his expertise directed at our collective genomics problems. We've generated very high-quality, hi-fi whole genome assemblies for over 100 diverse cannabis plants in our SeqWell2, derived from our internal breeding efforts and some key publicly available clones to build the library for initial pangenome construction. These include representatives of all chemotypes, 1 through 5, plants with pentyl and propyl cannabinoids, and what we believe to be the pantheon of potential photoperiod responses, unique phenotypical expressions like Australian bastard cannabis and fasciated specimens, and many diverse aroma palettes. Busco assessments of completeness demonstrate that these assemblies offer significant improvements over the previous reference. The irony of pangenomic promises to move away from a single reference standard is that doing so requires at least one good reference for bioinformatics packages to map new reads to. So while we've all been talking about cannabis pangenomes for the last 3 years, progress has been agonizingly slow. The current reference, CS10, is substantially more complete than previous publicly available assemblies and incredibly useful for many types of inquiries, but appears to lack concordance with more recent higher quality sequencing work. Kovalchuk's 2020 review of assemblies is a great starting point for understanding the legitimate debate about the nature of these issues, but the implications of subpar genome representation is critical for plant breeders. A great example of this can be illustrated by an alignment of chromosome 7 from CS10, both of which are type 3 plants, with one of our type 3, CS10 is a type 3, so is the plant that we are comparing to from one of our assemblies. Chromosome 7 is the physical location of THCAS, CBDAS, CBCAS, phytochrome E, several diterpene synthases, a propyl precursor gene, and a marker for overall potency. The different colored rectangles represent localized collinear blocks, which are conserved genomic regions between the two. This particular alignment suggests that the contents of each chromosome are similar, but also structurally divergent, which, if real and not an artifact of the sequencing and assembly process, would indicate widespread and recurrent rearrangements throughout. We suspect that this is attributable to the lower quality sequencing approach used on CS10, which was an older Oxford nanopore chemistry, as well as assembly artifacts that arise when collapsing two real haplotypes into a single synthetic haploid. So in other words, the two underlying haploid genomes of a diploid plant that are real get combined via software into one not real software representation of what the plant actually is. This ability to do diploid plants is a brand new opportunity. There's very few other genomes in terms of reference genomes that have been fully phased and released as both haplotypes. So we used We used several sequencing approaches to build a new pangenome anchor reference and brought in collaborators from PacBio, BioNano, Dovetail, NC State, Cornell, and Oregon State University. The plant serving as reference is the same F1 hybrid we self-pollinated to generate our segregating F2 breeding population, yielding autoflower and Varin markers, and has a genome size of 1.4456 gigabases, as estimated by Shen Chen at NC State. The two parents, one a type 1 high propyl 16-week flowering nightmare of a plant, and the other a day-neutral type 3 plant that flowers in about 6 weeks, were sequenced using PacBio's CLR technology. The F1 itself was sequenced twice using PacBio's HiFi process, generating 2 full smart cells of data. HiFi ASM using both CLR parental data sets as input guides. HiFi ASM was then used with the trio bin function using those 2 parental assemblies as input guides. Hi-C data on the F1 was generated by Dovetail Genomics and was used to scaffold contigs into pseudochromosomes for both haplotypes, and the resulting draft assemblies are now being verified against optical maps created by BioNano, and these are amazing. They're really cool. They don't give you any indication of nucleotide identity, but they'll tell you basically the full length and size of chromosomes. And we're also using linkage maps constructed by Dustin Wilkerson at Cornell, generated from NR gene Illumina shotgun reads of 289 of the F2 S1 individuals from our reference example. Dovetail is currently annotating both haplotypes using multiple tissue RNA-seq runs. So the project's nearing the finish line, and we're really excited to release this first phased diploid cannabis genome for everyone to use. But the initial results have already proven useful for us and allowed the deployment of some promising pangenome tools. Most importantly, the results are suggesting most cannabis chromosomes are fairly stable relative to what many of us previously believed. Here's the same comparison of chromosome 7 from the 2 haplotypes of our draft reference assembly, just to highlight the improvement experienced. Where the previous chromosome 7 alignment produced acid reflux, the alignment between haplotypes in our assembly is more like a sigh of relief. And that's my interpretation here, despite the fact that one is THCA containing and the other is CBDA containing. More technical details on this entire project will be released by a much more competent scientist who are working with us. We're shooting for a fall 2022 to an early winter 2023 release of all this data and the creation of the first publicly available cannabis pangenome. I'm almost done. Thanks. Says I have 2 minutes. We'll see here. Speaking of fall 2022, one last announcement. We're using this information gleaned from these all-to-all comparisons to help us design varieties that maximize heterosis. MASH is one of these pangenomic tools that estimates genomic distance and mutation rates within our plant library, giving us unparalleled insight into the relatedness of particular plants. Results from early analysis suggested that the inbred photoperiod line developed for our white CBG hybrid seed is the most genetically distant from others in our library. So to take advantage of that, we paired it with 2 CBD-dominant day-neutral pollen donor lines to create 2 new hybrid type 3 lines. We field trialed these in 2021, and we're really impressed with the results. They were standouts in the field for yield and flower structure. These positive results, along with our internal analysis of the underlying whole genome data, are aiding our breeding decisions as we move forward with commercial type 1 and type 2 varieties. Our first release of type 1 and type 2 F1 Triploid seed will take place this this fall, with a specific eye towards the THC flower market and a pairing of classic flavor architectures with exciting twists informed by our internal flavor development program. Initial tetraploid production mothers for these projects include a number of really interesting inbred lineages derived from ChemD, Triangle Kush, Durban Poison, California Orange, Trainwreck, Urkel, and some of our own internal lines as well. So we're really excited to pair these tetraploid production mothers with some special new day-neutral varieties to create basically the same type of award-winning hybrids that we've done on the hemp market, but bring them to the THC side. So while these advancements offer a lot of hope for the future of cannabis production, we're really stoked to make it possible. They're happening in real time and subjected to the whims of an unpredictable market, which fundamentally challenges every producer's right to exist on a daily basis. We hope that these plants will give farmers a fighting chance to realize their own dreams as well. And with that, we'll end for today and take questions. Thank you.
Hi, and thank you. You did mention at the last part of your closing here about the THC market. And does this mean that the triploid intention is not really available in the THC market?
It's currently not available in triploid form. That's correct. Yeah. About 3 months.
Meaning no one else is doing it either, as far as you know? Meaning no one else is doing it either, as far as you know?
That's correct. There's one other company that's working on it right now. I'm sure there's other companies working on it. It is the future, but in terms of being available in the market, there are none right now. Yeah.
Thank you.
Yeah.
Thank you so much for the presentation. Very, very nice talk. So my question is, I want to ask about the environmental suitability of the triploid. So it seems that the strain that you're starting with, in fact, no strain that we are growing right now in the US, none of them are selected for the growing environment in the US. I was just wondering, have you looked into the growth, the environmental parameter that would be suitable? I mean, that would fit the grow environment in the US. What is the range that you can possibly grow this triploid plant in the US? And if not, is there any interest for research to look into those parameters, like to figure out where we can grow this in United States?
I'm going to apologize. The audio is terrible up here. I can't.
I guess I just want to ask about the environmental suitability of the triploid. Very, very nice talk, by the way. I was wondering, have you figured out where we can grow this in the US? Like in terms of the temperature range, photo period, you know, like those.
Gotcha. Yeah, no, that's perfect. Thank you. You can grow them anywhere in the United States. The photo period response is similar regardless of whether you're in Vermont or Florida. So they're hybrids that have been designed in a very particular way to allow farmers to grow them and get them basically harvested and out of fields by early October. What you're specifically talking about, and I saw the previous presentation, it was awesome. You can basically grow industrial hemp, which is cannabis. It's the same thing. There's just a single gene difference between, in most cases, a single gene difference. You can grow them anywhere with a hemp license, so anywhere in the United States.
That was awesome. So I appreciate the whole thing. So I'm actually an environmental biologist. And everything you just said and the exact question that he just asked is exactly where I'm going with this. So I'm actually a part of the Midwest Hemp Database Program. We use Surfer Haze and white CBG, I think it is, as part of our Cultivar Check program. We send those seeds across the Midwest and grow them in different environments to this point. Have you all looked at any epigenetic data as to why some of these changes, because again, they are still all compliant, right? But they're showing different levels. Have you all looked into any different epigenetic stuff in those different environments?
So it's a really good question. No. No.
We'll talk afterwards then.
What's really cool, and it's worth mentioning, PacBio just released a brand new tool that goes along with their sequencing that allows for the detection of methylation in whole genome reads, which is opening up a whole, like this is like, Everything they've been doing has been revolutionary. This is the next phase in revolutionary sequencing. It's going to be pretty incredible. But to directly answer the questions in terms of different varieties, the problem that we ran into is that different varieties in different locations ended up producing some seeds in certain circumstances. So there were genotype-specific differences, but what we ended up seeing is that the genotype differences sometimes would change depending on which area that it was in. There weren't enough samples to be able to really pull that apart, so we do need more data. And we didn't see the same outcome in the university field trials or our own internal trials. So it's basically one of those unfortunate situations where you just have to say, we need more data to make a good claim. But at this point, there is something interesting going on, and we just don't know.
Awesome, thanks.
Thank you.
Hi, Sharon's my name. Curious question. The colchicine that you're using at the initial stage, is that the same anti-inflammatory drug?
Yeah, it's great for gout.
Right. And so are you using it as the anti-inflammatory to suppress, or?
In specific doses, it stops microtubule. I can't. I was going to pretend that I know what I'm talking about.
Well, don't think I'll understand
Basically,
either.
it stops chromosomes from dividing, stops cells from dividing, and so instead of getting that division phase, you basically just get a replication phase, and you end up with 4 sets instead of 2. So rather than an anti-inflammatory, it's actually at a cellular level stopping things from dividing.
The reason I asked that question is colchicine just recently, it's being used in cardiovascular disease, which is an off-market claim, and that's on the endothelial of the vascular system to stop that plaque replication. So it was an interesting, thank you.
Yeah, it's funny how certain things that are developed for one application can end up having a million different uses. And in the plant breeding world, it's widely used for stopping cell division. Yeah. Please.
Thank you for sharing such amazing information with the public. It's an honorable thing that you're doing. My question was about germination rates, and maybe I missed, did you have any issues with germination rates or failure to thrive with some of the triploids?
Yes, that is a great question. So what we've noticed is even in diploids, we have varying rates of basically conditioning that has to happen for certain varieties of seeds. Some are immediately ready. You can germinate them the week after they're harvested, and you'll get 100% germination. Others, you have to wait up to 6 months. They have to go through a conditioning process, stratification process. Triploids are more impacted by that. Tetraploids, even more impacted by that. We're seeing it in regeneration as well, taking cuttings. Triploids are much faster to basically clone. Tetraploids are a little bit slower, and as we go up the ploidy chain, the higher you go, the more difficult they are to work with. So most definitely a ploidy relationship between dormancy and seed, for sure.
Awesome. Thank you. Fascinating. Thank you.
Yeah.
Hi, really appreciate the talk. My question is building off the colchicine as well. Curious if you're seeing any other notable mutations following that, and maybe that might be involved in your environmental, epigenetic expressions that you're seeing too.
Yeah, that's a fantastic question. That was one of my biggest concerns. I've just spent the last 3.5 years developing this plant, line breeding it. Now you're going to put a mutagen on it and expect us to make seed with it. Get out of here, right? We have sequenced using the older CLR technology, we sequenced the first tetraploid that we made. Didn't see any differences, but I don't trust the older technology. We're in the process of getting sequencing data and doing the comparison right now through it's a graduate student project at Oregon State University, taking a cannabinoid-free plant that we basically gave the university to work with that went through the same process. And we have underlying sequence data from the diploid form, and now we're going to be doing the sequencing and comparing it. So far, we do see some strange things when it doesn't stick. So like if you have a reversion on part of the plant, you'll end up seeing phenotypical differences. But for the most part, there really isn't widespread mutations that people would sometimes think about, like with gamma radiation or something like that. It seems to be true to type phenotypically anyway, just better. Like if you have a diploid plant that's great, best way to make it better is to turn it into a tetraploid.
Perfect, thank you.
Yeah.
Hi. Over here. Fantastic talk. Thanks so much. My question is about the THC seeds that you referred to at the end. Do you have a distribution model set up that we could get seeds in California, for example? Or?
Yeah, go to gtrseeds.com, enter your credit card information, and we'd happily take you your money.
Fantastic. But seriously, from a regulatory compliance standpoint, if we're bringing in seeds into California that's coming from another state, how are we going to get that into our metric systems and compliance? Is all that going to be taken care of through interstate orders?
This is part of the problem, is that it's state to state, right? It's dependent on each individual state. The only thing that we're covering our backsides on is our legality to make sure that we can send them out. They're sent out as industrial hemp seeds, which are completely 100% legal in the United States. It's up to the individual cultivators or home growers who buy them to check with their local authorities and regulations before popping the seeds.
Okay, thanks.
Yeah, great question though, thank you.
Thank you for your nice talk and nice work. My question is related to the natural tetraploid plant that you isolated from Himalaya. Did you sequence the genome and did you mine for good alleles that could explain its resilience to this climate? You know, the resilience can be due to the doubling the number of genes, but also accepting mutations in the second genome without being lethal for the plant. So I was wondering if you mine for very good allele or...
And that was in reference to the Indian tetraploid population? No, and honestly, I haven't been able to get in contact with that group that did the research, and we've been trying for about 2 years. So if anybody has any way to get a hold of them, that would be great.
Okay.
But I'm very curious because it's such a unique population for all those reasons. It would be amazing to get the germplasm so that we could look at what makes it different.
Yeah, I'm sure it has evolved a lot, and the second genome has accumulated some potential beneficial mutations, and it will be nice to sequence this. Yeah. Also, do you have an idea why tetraploids haven't evolved everywhere on Earth? You know, if they're producing more, if they're more resilient, why the evolution didn't make this tetraploid to colonize Earth?
And I'm really sorry.
Sorry, no, I have a strong accent on the top, so.
I'm usually really good.
I was wondering why evolution didn't keep tetraploids instead of diploids. If they are growing better, having a...
In the triploid population?
No, why just tetraploids haven't colonized Earth?
Oh, God, that's a huge question. That's a really big one. And I'm not an expert in the area. I would defer to people who really do study polyploidy. My best guess at this, and from everything that I've gathered, is that you have more gene copies. It makes you more resilient in places where there's challenges. What we see is that a lot of the invasive species that really make headway across ecological regions are polyploids. And they end up, for whatever reason, being better at succeeding in those areas, which is super interesting because fertility is almost universally lower in higher ploidy plants. So they're making up for that lowered fertility with a better adaptability is what it seems like.
Balance between seed production and resilience.
Yeah, it's fascinating. We only have so many years, right? So you got to choose your investigations wisely.
Hi, thank you for your great presentation. I work in indoor cultivation. I'm just curious to know if you guys have studied using triploid plants in indoor cultivation, or if you have any plans to do so, and what kinds of advantages do you think there would be to using triploid plants indoors?
Using triploid plants indoors?
Mm-hmm.
Triploid plants and I would say tetraploid plants are dramatically superior to diploid plants. We just obtained a couple THC production licenses in Oregon, specifically in preparation for national legalization, and we don't grow anything except converted tetraploids in that facility. That's it. Triploids are great as well, but in terms of getting something true to type very quickly, the fastest thing for us to do is develop a plant variety at the diploid phase, convert it to a tetraploid, and then put it into production. And honestly, you have never experienced cannabis until you walk into a greenhouse full of triploids or tetraploids compared to a diploid environment. It's like the ultimate expression. We're about to flower the 8X plant, the octoploid. If it's better, I'll tell you next year, but so far so good. Yeah,
Great, thank you.
awesome. Thank you guys, really appreciate it.